Hello everyone, in this video we're going to talk about meiosis. You've all been doing a really good job getting your homework done and paying attention in class. Hopefully you'll do a really good job on the test on Friday as well. So let's take a look at the notes for meiosis. First off, I hope you understand what's going on in this picture. Starting with, another picture, as you can see, apoptosis. Okay, so let's actually get started. The first thing that you want to make sure you understand is the difference between chromosomes and chromatids and the number of chromosomes versus number of DNA. Okay, so before we get started with talking about meiosis, we already talked about mitosis, and with mitosis you have chromosome numbers that you can count. So when you look at the cell on the left, for example, you have 4 individual unduplicated chromosome. Okay, so what does that mean? It means each chromosome only contains 1 DNA, is unduplicated, and you have 4 chromosomes in total. So you have 1, 2, 3, 4. 4 unduplicated chromosomes. And then after DNA replicates during the S phase of the cell cycle, we come up with duplicated chromosomes. But we still have 4 duplicated chromosomes. So we have 1, 2, 3, 4. 4 duplicated chromosomes. However, since you've gone through DNA replication, instead of having 4 pieces of DNA, 1, 2, 3, 4, 4 pieces of DNA, you now have 1, 2, 3, 4, 5, 6, 7, 8 pieces of DNA. But 2 pieces of DNA are connected together at the centromere region of one duplicated chromosome. So this one duplicated chromosome has 2 pieces of DNA, 1 centromere, and this is 1 chromosome. Okay, 4 unduplicated chromosomes, 4 duplicated chromosomes, and each side you have something called sister chromatids. Another thing that you need to talk about is the concept of homologous chromosomes, and we'll talk about that on the next slide. So there are some notes to fill out over here. We're counting chromosomes. On the left side we have 4 chromosomes, 4 DNA as well, and this is a 2N cell because we have pairs of chromosomes. This is a diploid cell because we have pairs of chromosomes. Anytime you have pairs of chromosomes, that's a diploid cell. And then on the right side, we still have 4 chromosomes, but now we have 4 duplicated chromosomes, and now you have 8 pieces of DNA because you've gone through DNA replication. But this is still a 2N cell. This is still a diploid cell because you have 2 pairs of chromosomes, and again, anytime you have pairs of chromosomes, that's a diploid cell. We went from 2N to 2N. The chromosome number did not change, and it will never change during DNA replication. The next thing that students often get confused of is the difference between sister chromatids and homologous chromosomes. So let's take a look. When you look at sister chromatids and homologous chromosomes, sister chromatids are 2 pieces of DNA that are identical to each other, and they're attached, and they're on 1 piece of chromosome. So when you look at this green chromosome, for example, it has 2 sister chromatids. This is 1 sister chromatid. This is another sister chromatid. They're identical to each other. On the right side, this is another separate chromosome. 1 sister chromatid, 2 sister chromatids. They are exact copies of each other, but they're on the same chromosome. That's sister chromatid. It's 1/2 of the duplicated chromosome. They contain the exact same copy of genes. You have the centromere that allows the 2 DNA to be attached, and then they divide during meiosis too, which we'll take a look at in a second. And then we have something called homologous chromosome, which is different from sister chromatids, and you need to understand the difference in order to understand how mitosis and meiosis work. So homologous chromosomes are 2 duplicated chromosomes that contain the same gene. And there are 2 separate chromosomes, but they could have different variations of the same genes. What does that mean? So what you see over here, we have a pair of homologous chromosomes that are unduplicated. Over here we have a pair of homologous chromosomes that is duplicated. This is 1 pair of homologous chromosomes. There are 2 completely separate chromosomes, 2 X shapes, okay? And they can be paired up during meiosis 1, and then they divide during meiosis 1. But what's so special about homologous chromosomes? They're the same size, and they have the same genes. What does that mean? If we were to have eye color right here, over here has to be eye color. Over here has to be eye color. Over here has to be eye color as well. So homologous chromosomes contain the same genes at the same location. So if you have the eye color gene right here, all of these are going to contain the eye color gene. But then you can have different variations of the same gene. So this one right here could contain the blue eye gene. But over here, this could contain the brown eye gene, right? They are both eye color genes, but they could be two different variations of the same genes. That is homologous chromosomes. And whenever you have a pair of homologous chromosomes, in our bodies, right, we have 23 pairs of homologous chromosomes. And what are these, right? Your 23 pairs, 23 of those, this one set of chromosomes, 23, comes from mom. That's the genes that you're inheriting from mom. And then you have 23 that come from dad. Those are the genes that you inherit from dad, right? So whenever you have a pair of homologous chromosomes, one of the pairs is from mom, one of the pairs from dad, and you're made up of genes that are from dad and from mom equally. So you have homologous chromosomes, pairs of chromosomes that contain the same gene, versus sister chromatids, individual chromosomes that are identical copies of each other, and they're on the same chromosome. They're just duplicates, okay? Now, we can take a look at meiosis, starting with interphase. Just like all the cells, you have to prepare for meiosis, and you have to prepare for mitosis. So we have interphase, G2. By the time we get to G2, you had already gone through G1, and you had gone through S. And during the S phase, DNA replicates, right? So during G2, the DNA is already replicated. You have 23 pairs of duplicated chromosomes. And the DNA is uncondensed, so we do wiggly fingers. This is the interphase. And then we have nuclear envelope remains, because we're still at interphase. We don't need to release the DNAs yet. And then moving on from interphase, we enter prophase 1. So during prophase 1, the wiggly fingers become condensed DNA, so you can see the X shape, and they pair up, right? So we pair up homologous chromosomes. That's one of the events that happens during prophase 1. Something else that happens prophase 1. Well, the two events, right? We have DNA condensed, we have DNA pair up. We also have crossing over, and I'll talk about that in a second. And we also have nuclear envelope starting to disappear. So let's take a look. Are the chromosomes replicated? Yes. Chromosomes pair with their corresponding homologous chromosomes and form a structure called tetrad. You don't really need to know the word tetrad. But the point here is that each homologous chromosomes will pair up. Okay? So if you have 23 pairs, you go pair, 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 pair. 23 pairs. And then we have crossing over happens to allow new genetic combinations. So how does that work? You have two duplicated chromosomes. A part of the non-sister chromatids will cross over, and then they will switch a piece, okay? So if I were to show you using markers, this is how it's going to look like. So we have two unduplicated chromosomes. Now we have two duplicated chromosomes. Two duplicated homologous chromosomes, right? They pair up, homologous chromosomes pair up, crossing over. And because they're crossed over, some of the bonds are going to break. So we're going to switch a piece. And now, this is what you end up with, right? So although we started with chromosome that came from mom and only contains genes from mom, chromosomes that only contain genes from dad, now that you switch a piece, they're not so similar anymore. Now you have this chromosome that contains both genes from mom and dad at the same time, and so is over here. So that's crossing over. If you were to count the chromosome number in a human body, the number of chromosomes will be 2n, 46 chromosomes. 46 chromosomes, individual chromosomes, but it's 2n, so 23 pairs. And the number of DNA will be 92 because you've duplicated the chromosomes. You went from 46 unduplicated chromosomes to 46 duplicated chromosomes, so now you have 92 pieces of DNAs. And then you have 92 sister chromatids as well, because the chromatid number reflects the DNA number, okay? So that's prophase 1. Let's take a look at crossing over one more time. This is how crossing over happens. We have a pair of homologous chromosomes, they cross, they switch, crossing over. This happens during prophase 1. Now we enter metaphase 1. So instead of sister chromatids lining up at the metaphase plate, and again, metaphase plate is an imaginary region almost. It's a place where the chromosomes line up, but it's not a physical plate. It's not a tangible thing. So during metaphase plate, instead of sister chromatids lining up, we have homologous chromosomes line up. So what do we mean by sister chromatids line up? How would that look like? If you were to have a cell and you have all the X shapes in one line, and you have the X's being pulled away from each other, or the sister chromatids really, being pulled away from each other, this, what you see right here, is called sister chromatids lineup. I hope you can see it. And then, however, on this picture, what you see is homologous chromosomes line up. So instead of having X shapes all in one line, you have each pair being lined up. Okay, see the difference? Instead of lining up like this, you're lining up like this. So that's metaphase 1, homologous chromosomes line up. This is very important. This is what makes meiosis 1 different from mitosis, or meiosis 2. Okay, something else I didn't mention, spindle forms, right? This you should already know this from mitosis. The spindles form, they attach to the chromosomes so that we can pull the chromosomes apart. Homologous chromosomes line up across the center of the cell, or that's the metaphase plate. Now we can count the number of chromosomes. Still 46, 23 pairs, 2N, meaning this is still a diploid cell because you have pairs. Anytime you have pairs, that's a diploid. Number of DNA is 92. Everything is the same. Number of sister chromatids, still 92. Nothing has changed so far, okay? Moving on. During metaphase 1, there is another concept called independent assortment. What that means is, of course, you can have maternal on the top, maternal DNA, meaning DNA from mom, maternal DNA on one side, paternal on the other side, maternal again, paternal again, but you can also have maternal-paternal, but paternal-maternal, right? Such words, okay? So let's take a look over here. We have pairs of homologous chromosomes, right? Pretending this is one pair, this is another pair, right? We can have lining up like this at the metaphase plate. Let's say these are individual chromosomes. But you can also have lining up like this. This is when you only have 2 pairs of chromosomes. You can already have 2 different scenarios. But the number of possibilities only increases as you have more pairs. So you can have one pair like this, or this, or this, or this, or this. And it just continues. If you have 23 pairs, you have a lot of different possibilities of how the chromosomes can line up. This process, this lining up however you want, is called independent assortment. And it increases genetic diversity just like crossing over. And again, increasing genetic diversity is good because it gives you a variation of different types of individuals within the same population, which means if the environment were to change, or even if the environment doesn't change, the different individuals will have different chances of survival. They have the ability to possibly survive better than another individual. So having genetic diversity is always a good thing. So again, when you look at this picture, you can see how there are 2 different possibilities when you only have 2 pairs of chromosomes. Moving on, anaphase. So we've already had everything set up. Now we just got to split them apart. So now it's easy. Anaphase 1, the spindle fibers pull each homologous chromosomes apart towards the opposite ends of the cell. Chromosome number is still 46, 2N, right? Nothing's changed. We haven't split anything. We haven't split any chromosomes or chromatids yet. They're still in one cell. So the chromosome number doesn't change. DNA number doesn't change, 92. Sister chromatid number doesn't change, 92. We are not even splitting the sister chromatids at all, right? So this is anaphase. And then during telophase, none of the number changes. So far we started with prophase 1. The numbers hasn't changed at all. Then we move on to telophase 1. Telophase 1, the chromosomes continue to separate, and something else that happens is the nuclear envelopes start to reform, because in order for us to have 2 cells, you need 2 different nuclei for the cells, right? So nuclear membranes or nuclear envelope form around each cluster of chromosomes. The number of chromosomes is still 2N, diploid cell, 46 individual chromosomes, individual duplicated chromosomes actually. Number of DNA 92, number of sister chromatid 92, number still hasn't changed. Until we get to the next point, we have cytokinesis, okay? Cytokinesis is when the cell divides, and that's when the numbers actually change during meiosis 1. So here's cytokinesis the first time, because there are 2 times of cytokinesis during meiosis. Cytokinesis the first time. If you compare this to the last picture, right, we have 1, 2, 3, 4 different chromosomes. But after cytokinesis, you have 2 cells, and each cell only has 2 chromosomes. So we've divided the chromosome number by half. How did this happen? How are we dividing chromosome number by half? Because we lined up the homologous chromosome pairs and we're cutting in the middle. We're getting rid of the pairs so that each cell only has one set. There is no longer pairs. So here's cytokinesis. The cleavage furrow for animal cells, this is what it's called, where basically the cell membrane starts to cave in the middle so that we can divide the cell membrane and divide the one splitting cell into 2 cells. Or the cell plate, which is a growing cell wall for plant cells. The cleavage furrow or the cell plate forms to allow the formation of 2 new cells, and they're now both haploid. How did we go from diploid to haploid? We have 23 pairs of chromosomes again, right? And all the pairs are lined up in the middle. We cut it by half, right? And you can imagine this. I hope I say it this way. Hopefully you can remember. If we have 23 pairs of couples at the school for some reason, and we line them up all in this vice room, and we split them apart. And now you're going to end up with 2 cells, and you no longer have pairs. They're all singles now. That is what happens during meiosis 1, right? We have pairs of chromosomes. We'll split them apart. We no longer have pairs of chromosomes. But do you have to end up with 23 boys on one side and 23 girls on one side? No, because it could be boy and girl, but the next one could also be boy and girl or boy and girl the other way, right? So you're going to end up with random numbers of boys and girls on each side of the room, which is the same way as cells. You get a different number of chromosomes from that are from mom and dad ending up into in the meiosis, in the gametes that you produce during meiosis, okay? I hope that kind of makes sense to you. If you're not really sure, go find some people, move them around a little bit for some reason. Go to the library, find some people and move them around. Or if you're not sure, come see me, okay? Moving on. So cytokinesis, we are changing chromosome number now. Number of chromosomes now is 1. We went from diploid to haploid because we're splitting out the pairs. We went from 23 pairs to only 23 haploid because one set of chromosome. You only have one set of chromosomes now rather than 2 sets. But you still have 40, well, now you have 46 DNA, right? Because you cut the DNA number by half as well. And you cut the sister chromatid number by half as well. You cut everything by half. Okay? Let me give you another example. We have boxes of markers over here. This is one box of marker, right? It contains one of each color. So one box of marker. Very good. One box of marker. This is a haploid cell, okay? If we were to have a diploid cell, you have 2 sets of markers. You have 2 of each color, right? But during meiosis 1, what happens is each color is going to pair up with another color, right? You can kind of see they're all pairing up. All the colors are lined up, okay? And then during Anaphase 1, we're going to split up the homologous chromosomes. We're going to split all the way. So instead of having 2 sets of markers, now you only have 1 set in each cell. That is cytokinesis. Well, that's meiosis 1, okay? Moving on. Chromosome and DNA number is cut by half, right? That's meiosis 1. That's the most important. Once we get through meiosis 1, now it's easy. Meiosis 2 is basically the same as mitosis. So if you understood mitosis really well, you will understand meiosis 2. So now we start with prophase 2. We have 2 cells. It's already haploid. We have 2 chromosomes in each cell. The chromosomes each consist of 2 sister chromatids become visible, right? Because every single time you have prophase, the DNA has to condense. Number of chromosomes, N, 23. No pairs. We split the pairs. We split the couples. Everybody's single. Okay? Number of DNA, 46, because we have duplicated chromosomes. So each chromosome contains 2 pieces of DNA. I hope this all makes sense, right? Okay. If you're not really sure what's going on, back up and rewatch. Metaphase 2. Sister chromatids line up. Just like how we did for mitosis. Okay? Metaphase 2, the sister chromatids line up. Instead of homologous chromosomes, because we don't even have homologous chromosomes now. Everybody's single. So all the singles are going to line up in the middle. All the singles line up in the middle. That's metaphase 2. Sister chromatids line up at the center of each cell. Number of chromosomes, nothing's changing. We're just lining them up. Nothing's changing, okay? Number of chromosomes, 23. Number of DNA, 46, because we have duplicated chromosomes. Anaphase 2. Anaphase 2, you're splitting up. Now these human beings are not only single, they're also being cut in halves. It's a little bloody, but they're getting cut in halves. Okay? So they're single, and they're getting cut in half. It's unfortunate. Now we're on to Anaphase 2. What is going on in Anaphase 2? We have Metaphase 2, sister chromatids lineup. Now we're going to split the sister chromatids. We're going to split the sister chromatids. Went from X to no X. So when you're going from X to no X, you're doubling the chromosome number because you're going from 1 duplicated chromosome to 2 unduplicated chromosomes. Okay? So if you're okay. But in this case, in this picture, we have 2 duplicated chromosomes. We're going to 4 unduplicated chromosomes. What does that mean for a human cell? The number of chromosomes in a human cell will be 46. It's still haploid. Actually, yeah. You're going to 46, right? Because we're separating the duplicated chromosomes into unduplicated individual chromosomes. So you have haploid, individual, unduplicated chromosomes. It's still haploid because everybody's still single. You're just cutting them in half. They still don't have a pair, okay? Number of DNA is still 46 because you're not duplicating any DNA. You're not splitting up, or you are splitting up DNA from each other, but you're not splitting up the DNAs yet, so the DNA number stays the same. Chromosome number doubles because we're talking about individual, not chromosome pairs or duplicated chromosomes. 1 duplicated chromosomes, 2 unduplicated chromosomes. 2 pieces of DNA, still 2 pieces of DNA, okay? On to telophase. Telophase, you just keep pulling the chromosomes apart. You reform the nuclear membranes around the chromosomes. Number of chromosomes still 46, number of chromosomes still DNA. The only time we changed anything was the Anaphase 2. Cytokinesis the second time. Second time, you're cutting up the DNA number. You're cutting up the chromosome number, okay? Cytokinesis, they're forming cleavage foil or cell plate again. Your cells are still haploid. We're trying to make haploid cells, so we're staying haploid. Number of chromosomes, 23. So we went from 46 unduplicated chromosomes to 23 unduplicated chromosomes. Okay, see the difference? We're not splitting yet. It's still 46, or in this case 4. But now we split, so we have 23 and 23, 23 and 23, or 2 and 2 in this picture. Number of chromosomes, 23. Number of DNA, 23. Sister chromatids, 0, because in order to have sister chromatids, you have to have duplicated chromosomes. But right now you have half people, so there's no sister chromatids at all. Chromosome number and DNA number cut by half. Everything gets cut in half. Chromosome number get cut by half. DNA number get cut by half because you're doing cytokinesis and you're cutting the cell in halves. Okay? So let's do this overview one more time. We had diploid to haploid in meiosis 1. But meiosis 2, so this is a reduction event because you're reducing the number of chromosomes from whatever it is, pairs of chromosomes, so double the amount, to 1 set. 2 sets to 2 sets. 2 sets to 1 set, haploid, diploid to haploid, 2 sets to 1 set, okay? That's meiosis 1. But meiosis has 2 steps. Meiosis 2 goes from 1 to 1, haploid to haploid, okay? But you're going from haploid, duplicated chromosomes, to haploid, unduplicated chromosomes. Again, if you take a look at this picture, this is mitosis, where sister chromatids line up. Sister chromatids separate into 2 different cells, diploid to diploid. Nothing changes. Number doesn't change. The ploidity of the cell doesn't change. Diploid to diploid. Okay. Meiosis, however, if you look at it, the 2 parts, meiosis 1 splits the homologous chromosomes apart, so you go from 2N to 1N. And then meiosis 2, you're only splitting up the duplicated chromosomes. You're splitting up the sister chromatids, so you go from 1N to 1N. That is all for today. You can take a look at this chart that summarizes everything, and I hope this makes sense to you. There will be another video showing you how this whole thing works with pool noodles, and hopefully you'll take a look at that video as well. There will also be a video posted for mitosis. Take a look at that one as well. Good luck!